Welcome everyone. Welcome to our new webinar series, which is a format as a podcast. So today we have our first distinguished guest from Valutech, Greg Heland. He is the co-founder and the CEO of Valutech company. Welcome, Greg. How are you?
Thank you. It's a pleasure to be here and to participate in this exchange of information.
Thank you so much. And we spend a lot of time together, especially in IEST events. And we have been discussing about such projects that we can do together. I think it will be a nice discussion because we are always discussing about the particle part of the regulations and standards. Today we are going to talk about different parts. As you may know, Valutech is mainly active on cleanroom supplies, gloves, wipes, and cleanroom suites. And they have a waste of information about the clean rooms and especially all the consumables. So today I have a couple of questions for Greg, but before we start, I have one question as an industry trend. You have been in the industry for a long time. How many years, Greg?
I started in the industry right out of university in 1982, so I've seen a lot of change over 4 decades.
That's great because I think you are the right person to answer this question. My question is, how have you seen the cleanroom and contamination control industry evolve over the past few years?
As regards to the advanced material space, we've seen more isolation of the product in the process from the operator as the miniaturization and Moore's Law takes hold. And then on the life science side, there's really been an encouraging trend that more products are developed and tested in a controlled environment. And that's cascaded down to more guidance, more rules, really more GMP guidance in terms of best and recommended practices in the controlled environment. So I think to answer your question, since 1982, the cleanroom industry has exploded. And now with all these new life science applications, it's now more important than ever to be aware of the best guidance and the regulations that might impact your manufacturing scope.
Very good. Thank you so much. And what are some of the biggest challenges currently facing the industry, especially in contamination control area?
I think one of the largest overarching challenges is helping the operators to understand what I call the why. Technicians, operators, people that work in controlled environments want to do the right thing, but sometimes facilities get too bogged down with the checklist, must do A, B, and C. And it's really important as more people come into the controlled environment space that organizations build not just a protocol, but a culture around what best practices are, really to help the operators understand the why. If the operators understand why they're the rigorous protocols and guidance, you're much more likely to get guidance and you're much more likely to get compliance and consistency.
That's important, as you said, especially with the operators, to get them trained and understand the regulations and industry standards. I think that's the key. That's the most important part of our industry because things are changing rapidly and evolving a lot over the years, and understanding how they are reacting and how they are answering all these requirements in industry is based on their knowledge. And to gain this knowledge, of course, education is the key. We're going to discuss about European GMP Annex 1. As you know, it's recently updated. And at the beginning, I mentioned that we already covered the viable and non-viable contamination control, airborne particles and microbiology part of it. But there are some other parts that we never touched before on our webinars. So today we are going to discuss this part, essential parts of the GMP Annex 1. But before I start my questions, for the listeners who may not be familiar, can you please explain a little bit more about the European GMP Annex 1? What is it and why it is so important for our industry?
The European GMP Annex 1 is very important because they've taken the leadership position in terms of really what should be best practices as it relates to life science, specifically critical pharmaceutical, sterile and non-sterile applications. For decades, Europe has been recognized as kind of the leader in innovation as it relates to pharmaceuticals, and they've taken the lead with basically giving some coherence and compliance to, as the regulations and the standards have increased, having one central repository where you can go to this document to get clear guidance in terms of what are the regulations.
One important question, people always asking the same thing. As you know, Lighthouse is a US-based company, so the Valutech, and we are all working mainly for the US market. But when it comes to GMP Annex 1, this is European norm. Why it is so important for the US-based companies as well?
That's an excellent question. Since the globalization of the pharmaceutical and the life science marketplace, the interconnectivity between raw material formulation, testing, compounding, R&D, all these processes are done in potentially different continents, different locations, sometimes even by outsourced organizations. So the GMP Annex 1 affects the American market because it's hard to bifurcate. You can't just say, oh, this product, everything from the stern to the bow, it's not all done in North America, right? Because of the interconnectivity of the supply chain and basically how should we put it, how the disciplines, the highest and best use, there are certain organizations that have higher expertise. They might be in Europe. And it's important that if Europe passes this standard, that it be recognized as affecting everyone in the supply chain, because the finished end product that might start in the EU, be packaged in North America, and end up in the EU, it falls under the jurisdiction. So it's impossible to break this apart anymore. And that's largely because of the globalization and the interconnectivity of everyone within the supply chain, within the ecosystem.
That's a very important point. Thank you very much, Greg, because we all know that not only manufacturing in Europe and selling it to US or overseas, also products that are being manufactured in the US can end up sold in the Europe. So if you are willing to sell your pharmaceutical products, sterile form products in Europe, you have to follow the regulations. Plus, more than this, as you mentioned, FDA regulations, European GMP, they are very important, top-notch requirements for the industry. So if one company has all these requirements in place and gets certified by the FDA, by the European GMP, which means that they are able to fulfill most of international requirements. This is a bench top for our industry. That's a very important part. Let's go back to our original question set. As you know, the European GMP Annex 1 updated in August 2022, forced in 2023, and the partially lyophilization part of it will become available in 2024, in August this year. So there are a couple of things, especially with the packaging of the consumables, are generally mentioned as appropriate packaging. What is considered appropriate in the packaging of the consumables in the clean room?
Most important on the packaging, the first step is the labeling. The labeling should clearly identify who is the manufacturer, a lot number that's specific and traceable to raw material and data manufacturing, a country of origin, and an expiration date. These are all very important in terms of the identification. And then as it relates specifically to the packaging, clean room critical products should be packaged in clean, inorganic material. Organic material is known to carry endotoxin and potentially represent bioburden and viral contamination threat. So I just brought an illustration of whether your glove is a nitro glove or a latex glove or a PVC glove. It doesn't matter what the glove substrate material is. The appropriate packaging for a tissue for your nose is a chipboard box. A chipboard box which is uncoated because it's an organic material can and will support endotoxins, is not appropriate. So the key takeaway is that clean product should be packaged in clean packaging that will not potentially contaminate the product.
Taking one note, you mentioned that the cleanroom critical products. I think that's an important terminology, cleanroom critical products. What strategies can facilities employ to ensure cleanroom critical products are available once they need them?
The most important step is to be proactive, not to be naive to the stance that, oh, we've always done it this way and the plan A works and we're sole sourced. The IEST did a survey at the end of COVID, and it was uncovered that over 85% of facilities were adversely impacted by having a sole source strategy. So I think the most important thing is to recognize that you need to have a viable secondary option, a secondary supplier that you engage on an ongoing basis. And what a lot of our best-in-class companies have done is they've developed what we call a BCP. BCP is the acronym that stands for a business continuity plan. In bedrock in a business continuity plan is things like redundant power, what you do if there's an earthquake. But part of a robust business continuity plan is also this idea: what happens if the primary supplier can't ship the product? What is the plan B? And then actually having in place that qualified secondary supplier that's participating in the commercial benefit of supporting the project all along, so that if one path of supply is constricted, you can turn up the other path of supply.
Very good. Actually, there is one thing in the GMP environment, which is cross-contamination. So most of the things that we are trying to accomplish is to avoid this cross-contamination. And we all know that the main source of contamination is the cleanroom operators. So when it comes to cleanroom operators, their donning is the key to control contamination and avoid cleanroom cross-contamination between outsider area to the sterile environment. So when we take a look at this, what is the proper sequence for donning gloves to avoid cross-contamination?
The most important step is that the operator be in the right mindset. And let's back up a step to what I call the zones. There should be 3 zones to take in an operator from what we call street attire to appropriate critical environment attire. The first zone is what we'll call is the gray zone. The gray zone is an area where the operator should put in earplugs, a bouffant cap, safety goggles. Once they accomplish the gray zone, they should go to the blue zone, at which point, if it's a critical application, they don the donning glove. They don the donning glove after they put on a shoe cover. Then once they go from the blue zone to the white zone, with the donning glove, they grab their sterile garment with the clean glove to prevent the raw hand from contaminating the garment, and they don the sterile hood, high top booty, and bunny suit. And then once they've completed donning the garment and the face mask if they need to, then at that final point, they don the sterile glove with the assurance that there's no cross-contamination, because the sterile glove is the last component.
Thank you. I think that's important to follow these steps because the cross-contamination generally starts with the operator's poor gowning procedures. So if you're not able to follow the proper steps, then we will be able to introduce contamination within the clean room. And most people thought that we are bringing this contamination to the clean room through the HVAC system. In fact, the operators are the main source of contamination, and the poor gowning is the first entrance level for extra contamination to be intervened to our clean room environment. So also there seems to be some discrepancy about how many gloves an operator should wear depending on the GMP requirements, because we know that there are 1 or 2, or partially in the GMP it says, okay, the second glove should be over the first. And what are the best practice, and when is layering gloves appropriate?
That's a great question. Think of the setting and the application and the risk of what the operator is handling. There are a couple of pharmaceutical applications, radionuclear pharmacies, or facilities that are handling and compounding dangerous chemotherapy medication, where there's a real risk to the operator that if the operator comes in contact with any of the product that they're handling, that they could suffer adverse negative health consequences. So I think the easy answer is the first application is operators should wear two gloves in a situation where there's potential risk to them posed by what they're handling. The second application where an operator should wear two gloves is where it's a very critical sterile application and the potential risk of cross-contamination is high with the operator cross-contaminating the garment with the original donning glove. And so best practices are where it's considered a critical sterile fill, and oftentimes pharmaceutical facilities, they have perimeter clean rooms. They have an outer core and a middle core and then the inner core. So often the best practice is the operator has on the initial glove from the outer core, right? And as they migrate from the outer core to the inner core, they just never take off that outer core glove. As they move into the sterile area, they'll put on a fresh sterile glove before they go into what's commonly called the sterile core or sterile fill setting.
Very good. Actually, when we say gloves, we know that there are different types of gloves, and the ISO refers to the rubber gloves. What is this rubber glove? Can I use any kind of a rubber glove within the clean room? Is that okay?
No, that's not okay. The original term rubber comes from a derivative of what we'll call tree sap. And so not unlike taking maple syrup from a maple tree, a natural rubber or latex rubber is tapped from a tree. The FDA recognized over a decade ago that natural rubber is not acceptable in a controlled environment for a number of reasons. One of them is natural rubber has known proteins and allergens that are disruptive to a percentage of the workforce. And latex is actually a very dirty material. And so when people say use a rubber glove, really what they're referring to is what we call a NBR, a nitrobutadiene rubber glove. So NBR is a synthetic latex. And so just to clarify, natural latex is not recommended for a clean room. It's the synthetic latex, which is basically a petrochemical derivative.
That's important. NBR, nitrile butadiene rubber, right? That's
Correct.
a synthetic material. That's not a real rubber.
Right, and it's a little confusing that when somebody just says, "Oh, get the rubber glove," are we referring to latex rubber, which is natural rubber? Or are we referring to nitrobutadiene rubber, which isn't really rubber at all? It's the synthetic version thereof.
Very good. That's a very important point. Thank you very much, Greg. My next question is about the critical cleanroom products, reevaluation and testing. How often should critical cleanroom products be re-evaluated and tested?
Think of three primary hurdles that should trigger a reevaluation. The first one is a change in the production environment, right? Are we manufacturing a new product? Has a significant process changed in the production flow? Because gloves and wipes and apparel are all part of the operator and ensuring the operator doesn't cross-contaminate. Any significant production control in the environment should trigger at least a reevaluation of, hey, are the gloves, wipers, and apparel still compatible with the new production requirements? The second requirement is whenever you need to qualify a new manufacturer. When the decision has been made, be it through what we mentioned before, a business continuity plan, that an organization decides, hey, we need to have a plan B. We need to have a viable secondary qualified vendor. Well, at that point, when you're qualifying a new manufacturer, you're going to go through, that's going to trigger the step. And then the third one is often a little bit more difficult to discern. But when organizations observe what we'll call a variability in change, something changes with the performance characteristic of the glove and the wiper. It doesn't behave the way it used to be. It appears to leave contaminants. Operators will often observe these changes, and if operators observe a change in the consumable, that should trigger a deep dive in terms of reevaluating and making sure that the product being purchased is still of the same quality of what was specified. I will just make as a final comment that we often struggle with this. Me being in the industry for decades, we see a real complacency that because it's such a difficult process, it's so arduous for a facility to qualify a new supplier, there's often a hesitancy. People will sometimes, what we say in America, we say they push the can down the street, right? It's like, well, we've observed changes, but we can work around those changes.
So the next question will be about the test methods. What are some of the testing methods and equipment used to verify material cleanliness in a controlled environment? Are these internationally standards?
So the IEST is recognized as the organization that actually develops a lot of the test methodologies. And the intent of IEST, because it's a cooperative, collaborative organization, is to identify test equipment and test methodology that's readily available, that can be readily reproduced in a facility's lab. I'm going to throw at you a lot of acronyms because this test equipment tends to have complicated names. So the first one is something we'll call an APC, air particle counter. The second one is an LPC, a liquid particle counter. Both of these types of equipment use lasers to detect particle size and density. Another common test method is NVR. This is a test where you are determining non-viable residue. More recently, and we didn't use scanning electron microscopy or SEM until the last decade or so, that's used now in some very critical advanced material applications where they're concerned with certain elements, certain contaminant elements that can really only be detected by SEM. And then I also want to mention FTIR. So as you can see, there's a whole host of different test methods, and they all converge on basically determining the cleanliness and the appropriateness of the consumable to perform in the operator's environment.
Yeah, we also talked about the gloves and we discussed about most of the consumables. But one thing is important in new GMP Annex 1, which is SOC. And let's talk about it as well. According to European Union Annex 1, the SOX requirement, how does this rule affect North American pharmaceutical manufacturers?
Well, let me illustrate by just doing a simple exercise of gravity. I'm going to release my pen, and you can see where does the pen fall to the floor. So the root cause for the importance of the GMP Annex 1 as it relates to SOX is it's a well-documented fact that within the controlled environment, arguably the cleanest areas are at the ceiling, at the source of the filtered air, and the dirtiest areas are at the floor. And operators are known to take buses to work, ride bicycles to work, potentially walk across creeks and get their shoes wet to go to work. So the way an operator gets to work poses great risk to the cleanliness of the shoe and the sock. And as a result of a lot of study, it's been uncovered that if the operator doesn't take out that sock that's been worn in the outside environment, that's subject to gravity and all the contaminants that they run into shuffling about, that there's a huge risk that the operator's socks, their external socks, are they're going to introduce known potential contaminant source into the critical area. And so that's really the root cause for why to have clean room socks, to prevent that risk.
Exactly. I think socks are the most important part of our gowning procedure. In fact, this is the less uh, concentrated part also because people are just coming to the office by the regular socks, and we're always thinking about gloves and hair nets, all other stuff to put them into clean room, but we're not always thinking about the socks that they are going to wear within the clean room. That's why it's important. I have one final question. We mentioned IEST, and we also have some other industry regulations, technical reports, working group reports. Sometimes we have the monographs from different organizations like PHSS or ISPE, PDA. And also on top of this, we today discuss about the GMP Annex 1. And we know that the FDA has its aseptic processing guideline too. And top of all these items, we know there's an ISO standards. So how you see all of them? Are they working all together, or some of them are more important than the others? So how we can correlate between ISO regulations like a GMP, ISO standards, regulations like a GMP, or the industry documents like an IEST recommended practices? How we can use all of these documentation?
That's a great question, and I think a thorough answer might exceed the time boundaries of this podcast. I think historically, if you look at where the standards came from, the IST in the 60s was the originator of the original federal standard 209. The last revision was 209E, which basically those are the standards that people are commonly aware with, the class 1, class 10, class 100. However, the IST failed to update and revise those standards. And so really now ISO is recognized as the global standard, the international standard, if you will, in terms of the overall clean room environment and overall guidance. And so clearly there's some bias, and because ISO is more heavily weighed by EU, you might get into some politicking, but I think the fact remains that ISO is the gold standard for clean room overarching standards. IEST definitely plays a role. ISO documents reference IEST specific recommended practice documents. For example, I'm the chair of the working group for IEST RP CC005, which is recently going through a revision on how to test gloves. So think in terms of the hierarchy that ISO is the overarching cleanroom standard, and they will reference IEST specific documents in terms of how you achieve regulation or compliance within that specific document. You mentioned the FDA. The FDA closely regulates life science products that come to market in the United States. And so there are certain manufacturers that have the compliance requirements of the FDA. I think the best answer to your question in a short version is if a facility wants to have the best outcome, they need to be very curious in their space. They need to be willing and able to do a lot of discovery. They want to have the attitude that it's not about just checking boxes, it's about understanding how these organizations are related. And yes, there is a relationship between them all. And some of them have a focus in one area, and some have a focus in another area. So I think it's often difficult because sometimes people just want the easy answer. And regrettably, there's not an easy answer, right? There's many different compliance standards that you might need to adhere to based upon the specific manufacturing setting that you're in.
Very good point. Actually, when we are applying this to our industry, we generally say the same. ISO is for all industries. So, for example, ISO 14644-1 cleanroom classification, classification standard is for automotive industry, for aerospace, for pharmaceuticals. So you cannot really find the answer of your microbiological activity within the clean room, or you cannot really find any reason of your lithography room malfunctions within these ISO standard. However, industry-specific regulations are more depth and written by the subject matter experts from the industry. So we have to go and check what is written in these regulations to understand more deep. I have one final question where I can ask before we finished up our time today. I should say I really enjoy, by the way, this podcast. Thank you very much so far. And the future outlook. I have my final question about the future outlook. What do you foresee as the long-term impact of the new Annex 1 updates on the industry?
I think that that's going to create more forced collaboration between North America and EU. I think that at the long term, the outcome will be good because there will be adoption be between North America and EU. I just think the interim, the short term getting there, how should we put it? It's a little clunky, right? Because there's compliance issues. There's, oh, well, who from the EU is going to come in my facility in Indiana and create hardship for me? I think there's some people that are sitting on the sidelines. They're just kind of waiting. They're saying they're not refuting the science that the Annex 1 makes sense. They're kind of sitting on the sidelines waiting in terms of who's going to force me to act. Once we get through that awkward phase, which I hope is shorter versus longer, I think at the end of the day, it's going to be a really great outcome for the industry and then ultimately the consumers of these products. Because the reason why we're so interested in contamination control in life sciences is to make sure that we have products that don't unintentionally harm people, right? The whole reason for all the investment in life science is to increase the quality of life of people. And how can you do that with a product that has a potential cross-contamination risk?
Exactly, and GMP will also improve the way we look to the product and the way we protect our patient at the end, because whatever we do with these regulations are to ensure that the patient's safety is on the top and also the product is safe enough for our patients. Hey, Greg, thank you very much for today's session. That's the first time we tried the podcast session with you, and I really enjoy. Thank you very much for your contribution. And I hope we can come together soon, maybe face to face in the US or in Istanbul, so we can have the second part of our podcast face to face. Thank you very much for your time and also for your contribution.
Thank you. I appreciate your interest.
Thank you. So everyone, thank you so much for joining us today. We discussed about the GMP Annex 1, the new Annex 1. We have been discussing about the viable and non-viable contamination generally as Lighthouse worldwide solution. And we have a very important guest today explained us about the other part of GMP Annex 1, the consumables. So we learned a lot from Greg today, and I hope we can meet on our next session. Wish you a best. Good luck. Take care.